Welcome, and we're going to talk today about ending Parkinson's, and this is really based on a book that we have recently published, and the book is about Parkinson disease, and it's about the pact. So the ways in which that we can address this thing and really get it under control. It's a fun read, and it's much like if you like books like Malcolm Gladwell, if you like books that are that tell a story. We tell the story of polio and HIV and breast cancer, and we really get into the heart of a lot of interesting things. I do want to say before we start that there are 4 co-authors on this book. Ray Dorsey is the lead author. He's at the University of Rochester in New York, and he is an expert on telehealth and on care for patients, and is an outstanding researcher, and has one of the Parkinson grants called a Parkinson UDOL that looks at telemedicine. And so really cool on this project, because as everybody knows with COVID, we're doing a lot of telemedicine. Bastian Bloom is another one of our outstanding co-authors, and many of you know him. He's in the Netherlands and is just a world-renowned researcher, but also someone who has really taken it to the next level for developing care systems, and has developed the PD Net care system in the Netherlands, and has been deploying care systems around the world. Todd Scherer is one of our co-authors, and Todd was known when he was at Emory for working on pesticides and exposures, and so Todd has been a really great addition, because not only does he bring his expertise as the CEO of the Michael J. Fox Foundation, but he also studied pesticides and environmental exposures, and so he brings a tremendous amount of information and a wealth of knowledge to the project. So each of the co-authors is going to take a section of the book and present it to you in this series of webinars. And my section is on the treatments, and I'm at the University of Florida. I'm also the medical director of the Parkinson's Foundation and the executive director of the Fixell Institute for Neurological Diseases. I don't take any money from from industry sources, but I do have medical grants and research grants from a number of places, including the Parkinson's Foundation, the TREAD Association of America, Tyler's Hope for a Dystonia Cure, and the National Institutes of Health. And so I just wanted to make sure that you all know that, and then also mention that all of the proceeds from this book are going to go to Parkinson charities. So all 4 of us have made that commitment. So let's talk about the commitment, and just to introduce it a little bit to you. This is a picture of the book that came out just this month. It's Hachette Publishing House, and their sub-publisher is Public Affairs, and it's a great, fantastic read. And we're going to talk about this pact. And so I'm going to come back to it, because we're going to be asking people to take the pact for Parkinson's. Now, it's a little bit crazy because it came out in the middle of this COVID thing, so we lost some of our message. And so we're really trying to make sure that the word gets out about the book. And so if you have the book or you're interested in it, click on Amazon and write about the pact, the Parkinson's Pact. And if you have social media, write about the pact, and we'll teach you about that tonight. And the other presenters, if you're interested in their web-based seminars, they'll also cover this as well. And so it's so important that we all understand what we need to do to get to the end. Okay, so the pact is P for preventing, and you can see here there's a nice cartoon drawn by an artist from the Netherlands, and that's me holding it. And I'm not happy because I'm giving this prescription, but we're not where we need to be. The A is advocating, the C is caring, and the T is treating, and the T is the topic that I will cover tonight. I also want to alert you that we have a blog, and so this is my personal blog that I've had for many years with all of our books that we've published on different treatment tips and things with Parkinson's. And there's a piece on the blog about the pact. And so this is at parkinsonsecrets.com. So let's talk about the problem. How big is this problem? And it turns out that it's big. Parkinson's disease is now the fastest growing neurological disorder. It's growing at a faster rate than even Alzheimer's disease. Let me say that again. How big is the problem? It's big. It's really big, and it's rapidly expanding. And so this is something that we really need to get a hold of. Now, why do we care about this? Well, we care because if you look here at this graphic, it turns out that 1 in 15 of you, 1 in 15 people will get Parkinson's disease in a lifetime. Just pause for a minute and think about that. Think about your grade school. Think about the fact you had about 30 people in a classroom. And now think about 2 out of those 30 are going to get Parkinson's disease over a lifetime. It's really just an astonishing statistic and something that should draw our attention. So what's the next step? I mean, what is the next step for Parkinson? What do we do? Well, we propose in the book, and so the book is written where we tell stories and we talk about how other diseases have addressed this and been successful, more successful than we have been in Parkinson's disease. We tell the story of polio, the March of Dimes, and the drive toward a vaccine. We tell the story of HIV, and we talk about how they were so amazing in their advocacy, how they took over the National Mall in front of the Washington Monument, and they occupied the FDA building, and they put a condom over one of the Congress members' houses. And so they were active, they were aggressive, they were charismatic. They really agitated, and that's what's necessary. And so when we think about what the next step is, we have to study these diseases. And that's what we did in this book project. The 4 of us studied these diseases, and we looked at where successes have been and said, how can we apply this to Parkinson? And so this is why we want you to take the pact. Prevent the disease. Advocate, agitate, be charismatic, get out there, care. We have to care. We have a generation of people and a coming generation of people, and these numbers are exploding. And we need to begin to care for the people that have this disease and understand that if we don't develop better care and better treatments, that we are going to bankrupt many of the medical systems around not only the United States, but around the world. And then, of course, treating. And we're going to talk about the need to bring new treatments to the table. So how would I grade our response to Parkinson's disease? Well, I would say it's pretty disappointing. And why? Well, the disease continues to grow unchecked. Some people, some experts have referred to this disease as a pandemic. So pan is a Latin Greek derivation. Pan means all, okay? And demos is Greek for people. And so all people, all continents. So if you have a pandemic, we're thinking about all people, all continents. We're thinking about men. We're thinking about women. We're thinking about something that is growing. And so it does actually, from a Merriam-Webster's dictionary original derivation of the word fit for pandemic. Now, we don't use that word, and I'm not sure that I suggest using that word now because, we now associate that with infectious diseases. And so this is different than an infectious disease, but the ramifications can be much the same. And so this is growing rapidly no matter what you term it. And so how do I grade our response? It's been disappointing. So tonight we're going to talk about emerging treatments and research, and I'm going to present a few of the things that people are waiting for. One of my favorite visuals is a man standing on a globe looking at his watch. And I often say that when I treat patients, I think about that. I think a lot about that particular image because with that particular image, we are thinking about, we've got to hurry up. We've got people suffering from this disease. What are we going to do about it? So we need to move faster because there is a need for this. So when we talk about research, we talk a lot about the funding. And it turns out that Parkinson's disease is underfunded. It's easy for me to just throw that out, but we actually researched this and looked at the numbers and review the numbers in the Ending Parkinson's Disease book, and they are staggering. So let me just give you an example that may illustrate this for you. And my example is HIV, human immunodeficiency virus. I don't think anybody would dispute that this has been a major issue. When I started my medical career, if you got a diagnosis of HIV, it was pretty much game over, and it was a terrible disease to have. And we have put $3 billion a year through the National Institutes of Health, which is the largest funder of medical research in the world. We rose to the occasion and we put $3 billion a year into this disease, and we prevented thousands, if not millions, from ever developing HIV. That's a pretty good idea of the type of commitment that we need to have when we're agitating, when we're going after the FDA, when we're marching, when we're doing the A in the pact and advocating. And so Parkinson disease in 2019, 200 million a year. 200 million a year. That means it's more than a factor of 10 behind where it needs to be. So if we look at this across different diseases, and I really don't want to belabor you with too many statistics here tonight, but I do want to show you NIH funding comparisons. You'll see Parkinson's disease, I've circled there for you, and look at the funding, the NIH funding and the funding by the largest foundation doing research in Parkinson's disease, the Michael J. Fox Foundation. And if you look at funding, okay, research funding, look at arthritis, look at breast cancer, look at Alzheimer's disease, look at diabetes. We are just not going to get it done or get it done anytime soon with what we're doing. So why should we fund Parkinson's disease research? Well, here's a picture of the most common pill. This is called Sinemet. Sin means without in Latin. Emet means vomit, because you've got to have two components to it, the carbidopa, levodopa. So you don't vomit when you take it, but it's still the best treatment for Parkinson's disease. It's still the gold standard. Dopamine replacement pills are the best treatment for this disease and remain the best treatment. But the treatment, guys and ladies, it's over 50 years old. It's over 50 years old. So the treatment also does not stop disease progression, okay? And so we are left with trying to develop something that can address symptoms for patients, both the motor symptoms and the non-motor symptoms of the disease. And some people have called Parkinson's disease the most complex disorder in human medicine, period. Okay, and I don't say that lightly, but when you think about 20 motor and non-motor symptoms, and you think about all the different types of medicines and surgeries, when you start to think through all of these things, it becomes very complex very quickly. Oh, and then by the way, you take this pill here that's shown on your screen with yellow dye number 5 or number 6, giving it its color. That's Sinemet or carbidopa, levodopa, or Matapar if you live in Europe. And it may help some of the symptoms, but not all. So why should we fund? This is why we should fund it. We are not there with adequate treatments. Now, there are all sorts of cool new things that we're beginning to look at and use. And so this is a very cool, complex graphic from a recent paper by Ahmad El-Kouzi, who is one of our old fellows in movement disorders, who's at Southern Illinois University. He published this in Nature Neuroscience Reviews. And what he's showing here is this very complex circuitry. So these are all islands in the brain that you see. And these are all potential drug targets, new drug targets for Parkinson's disease. Some of the old, but a lot of new targets in the serotonin family and some of the family of things that we call NMDA receptors. There are all sorts of areas that we can target. And so a whole developing entire discipline is how we can begin to understand all of these new targets in the brain, including cannabinoids as well as another new target, to develop new treatments for Parkinson. And so I'm here to tell you there's a whole bunch of research that's going on in this area. Now, when we talk about drugs, we need to talk about this idea of slowing the disease down. Now, let me just back up and say, and we've said this ever since we put out the book called 10 Secrets to a Happier Life, we like to explain this to patients and to families in a very simple way. There are 3 buckets. Bucket number 1 are powerful symptomatic treatments. Those are like medicines and deep brain stimulation. Bucket number 2 are things that may slow disease progression in some meaningful way, or they may be something that changes the brain and protects cells from dying, okay? So that's bucket number 2, slowing disease progression. Bucket number 3 is the cure bucket, the C bucket, and that's the one that we're furthest away from. But if we can modify the disease, if we can slow it down, then you may end up having issues with other things besides the Parkinson's, your heart, your lungs, and other things. And so this is a great quote by Franklin Roosevelt, who says, "One thing is sure, we have to do something. We have to do the best we know how at the moment, and if it doesn't turn out right, we can modify it as we go." Disease modification is super important, and that's why we're searching for drugs that might slow the disease down and allow us to really extend the lives of patients without disability. And this could be an amazing advance. Now, just recently, meaning just this week, the isradipine study came out. This is a multi-million-dollar study from the National Institutes of Health and the Parkinson Study Group. The study was negative for slowing disease progression. Same with the inosine study and the pioglitazone study. These are called repurposed drugs. So isradipine is a hypertension drug, inosine is a gout drug, pioglitazone is a diabetes drug, and there's another diabetes drug that is very promising called exenatide that is in trial now. And then I also threw the anti-malarials on. And why did I do that? Well, it turns out when we look for drug hits in Parkinson, we also see those, and you've seen those in the news, as RNA drugs for COVID. So just kind of an interesting spin as we talk tonight. How about precision medicine as the next avenue of where we could go with Parkinson? Well, there's a lot of groups and a lot of researchers that are working on this area. Many of them focus on 2 of the most common genetic gene defects with Parkinson's disease. So this is basically changes in the DNA, okay, that are leading to Parkinson's disease. And these changes in DNA, all of the genetic causes, if you put them all together into one pot, they make up about 15% of all of the Parkinson's disease. And so we still have 85%, and some people would go up or down by 5% in those estimations, but we still have 85% of patients that we don't know what causes it, or it could be a gene plus environment interaction. So when I say precision medicine, what I want you to get excited about is we look at some of these genes across groups of people. The GBA gene causes a disease called Gaucher's disease. And if we look at that disease, there are enzymes that are important for it called GCase, and how they interact with alpha-synuclein, as one of these researchers is looking at it in a recent article on Molecular and Genomic Medicine. And so if we could target specifically these particular genetic abnormalities and get into the pathways, understanding what those pathways do, this is a great approach. And some people are even suggesting gene therapies, direct gene therapies for some of these diseases or for some of the symptoms. Ambroxol is one that's gone through safety testing and recently been shown in the journal JAMA Neurology to be promising potentially for people with these mutations. Another one is the LRRK2 or LRRK2 mutation. This is the most common gene for Parkinson's disease. Now, this gene exists in a lot of people. It's very common in Berber Arabs and in Ashkenazi Jews, but not just those groups of people. Sergey Brin has this gene, who's the founder of Google, for example, and you can see a nice pictorial of what that gene, the molecular structure, what it looks like. And when we have now studied LRRK2, we realize that we may be able to use drugs that inhibit what is called the LRRK2 kinase, which is an enzyme, or there's also efforts aimed at potentially gene therapy. And so this is a really interesting area we call precision medicine, because we're going specifically at these genes. And there are studies both with the Parkinson's Foundation and the Michael J. Fox Foundation where we're beginning to ask people to go ahead and get genotyped to get ready for the next generation of therapies. So if you have one of these may be the first things that we see treated by precision medicine. Now, how about deep brain stimulation? We often talk about sticking a straw into the brain and pushing a little bit of electricity in there, and that's what my lab has been doing for a couple of decades now, and trying to develop these new next generation ideas for this. And we are seeing a lot of innovation in this field. So we're seeing new devices. We've even seen a few new device companies come on the market with new approaches, and tons of researchers all over the world looking at different ways to push electricity into the circuits. So there are all these islands in the brain, and they're all talking to each other, and we try to push electricity into those islands and change the way they're talking to each other. And by doing that in some very amazing way, we can actually modulate, or sometimes we call it neuromodulate, and improve the symptoms. This has been probably the next greatest advance in Parkinson's disease treatment, next to that yellow dye number 5 or number 6 pill I showed you called carbidopa, levodopa, or Sinemet, or dopamine replacement. On this slide, I don't want to overwhelm you, but this is another slide from this beautiful review by Dr. El-Kouzi. We're showing typical electrical modulation, so we can stick those straws in, and you see 4 little blue contacts there on the left side of your screen, and it says invasive underneath invasive, and that's typical DBS. But now, with Ed Boyden and Nir Grossman and their group from MIT, we're beginning to aim electrical fields from outside the brain called temporal interference. And so we've begun research in this area as a non-invasive way to try to do this. It's not quite as good as DBS at this point, but it's pretty promising because you don't have to go into the brain. And so that could be something that develops. And then we're developing optogenetic approaches, meaning pushing light into the brain and genes into the brain to be able to study the brain and actually deliver pulses in sub-milliseconds in order to modulate these circuits. We've done this mostly in animals. And then we have some exciting therapies where we can push infrared light into the brain from the outside and try to modulate nanoparticles. And so a lot of cool things going on. One of the things that our laboratory does quite a bit with, along with Dr. Foote and Dr. Gundes, who are 2 of my collaborators here at the University of Florida, is building these next generation devices. And so if you hear the term closed loop or adaptive stimulation, what we do for this therapy is we actually in real time read out the brain signals that are coming to us, and then in real time, almost like an EKG, we can see those and we can react to them and teach the devices to react to them. And that's why it's called closing the loop. So before it was just called open loop. We would just openly throw electrical stimulation into the brain. And then it worked. That was cool. But now, if we can actually read the signals for specific symptoms, which is what we're interested in, and many laboratories around the world are interested in this. Helen Bronte Stewart and at Stanford, Phil Starr is doing a lot of great work at UCSF with Jill Ostrom, Peter Brown at Oxford. The list goes on. This is very cool. And we call this adaptive as well, so that we're trying to adapt to this individual symptoms for individual patients. And so this is a really cool therapy, and I think you'll be seeing more and more about that. So talking about horizon, what's on the horizon, I wanted to show you a couple of pictures. So you can see a picture of a mouse from Stanford, and this is optogenetics. And what optogenetics is it combines genetic engineering, optics or light, and light-sensitive molecules, and we can achieve precise control of cellular activity. And why is that important? Well, we're trying to understand what precisely is going on in these circuits so we can build the next generation devices, okay? And so oftentimes we'll use optogenetics in the laboratory, and then when we develop the actual deep brain stimulation or neuromodulation device, we call that optogenetically inspired deep brain stimulation, because optogenetics doesn't yet scale to humans for a number of complex reasons. And so maybe one day, but not right now. The other are DREADs, D-R-E-A-D-D, and that is a mnemonic that stands for designer receptors exclusively activated by designer drugs. Designer receptors exclusively activated by designer drugs. You can see this for a cancer drug in the picture in front of you from Genentech. This is just one of many examples. We're beginning to do this in the animals as well, and so we're beginning to develop DREAD-inspired deep brain stimulation and hopefully DREAD-inspired drug development. And so I really want people to know that there are some exciting things that are coming along. The other thing that's so important when we talk about emerging treatments is we must develop a biomarker. And so what is a biomarker? And you just hear this term thrown around, you know, just people just almost carelessly throw it around. Biomarker, I have a biomarker for this, I have a biomarker for that. We have to have a reliable biomarker for Parkinson's disease so we can measure the changes in disease activity. Why that is so important is because if we're going to do a drug study on Parkinson's disease, we're going to need thousands of people, okay, for drug and device studies. And this is going to take a very long time. But if we can develop a marker where we can see when we're actually changing the brain, changing the course of the disease, if we can develop a marker of that, a biological marker, you see where that word comes from, of those changes, and we can measure the progression, we can go from needing thousands of patients to needing hundreds of patients, cut down our development time, and at the same time, as we start to throw more money, remember we want to go from 200 to 300 million to 3 billion, we want to go more than a 10 times factor. The development of a biomarker is super important. So although I'm telling you about all these really cool things, I'm going to back up and say, man, if we had biomarkers, like this is a picture I borrowed from David Valancourt at the University of Florida. He's got several National Institutes of Health studies going on these biomarkers and a recent collaboration with the Parkinson Study Group. These are going to be important. Could be images, could be blood, could be spit, could be all sorts of things, could be skin. So you're seeing people look at biomarkers, so important for us. Now, a biomarker can be used either in some cases designed to diagnose a disease, okay, and in some cases to track disease progression. But for next generation therapies, it's probably more important for us to be able to track the disease progression in a very measurable way. Finally, I wanted to talk a little bit about immune therapies. You hear about this a lot in cancers. We don't talk about it as much in Parkinson's disease. The field has been stuck on stem cell therapies for too long, and we know now that stem cells are cool. We know that we can turn a cell into the brain from the skin into a brain cell. We can turn cells into dopamine cells. Very cool. But it's not so easy to just stick a cell, whether it's a stem cell or a cell from a fetus or a cell from yourself that's been repurposed. It's not so easy to just stick that back in the brain and have it do what you want it to do. And so stem cells have not really panned out to be the amazing panacea of what we thought they might be 15 years ago. However, one thing that has gotten very interesting is the idea of the immune system. So we talk about vaccines being the tugboats of preventative health, and this is William Foch, a great quote. And when we think about this idea that maybe we can teach your immune system what's abnormal, whether it's this bad protein called alpha-synuclein or something else upstream, and teach your immune system to go after that. And so we can develop not only vaccines, but immunotherapies. And so these are the things I wanted to review. I wanted to tease you a little bit about the book. I want to try to get you to commit to the Parkinson's Pact. I want you to take that pact. And remember, here's my prescription pad. And I'm going to write on that pad, we need to prevent, we need to advocate, we need to care, and we need to treat and develop these emerging treatments. And so the book has been out now for a week or two. It's hurting a little bit for its message. We really wanted to get this message out worldwide because of the COVID crisis. So we're depending on all you all to carry the message. The best way you can do that for us now when you're in your homes is just read the book and tell your friends about the pact. And if you do have time, we would love it if you could get on Amazon and give us a review and talk about the pact and the review if it's something that moves you, because we really need to get the word out. And remember, all of the proceeds are going to charity. So with that, I am going to stop my screen share here. I'm going to get right back on here and back in the studio here, and we are ready to take some questions. If anybody has any live questions that you would like to send me, if you look at your screen, there is an area called chat in the bottom of the screen, and so I'll give you a sense of that. And so while you're pulling up to chat, I would like to actually read an excerpt. I actually have the book here, and I want to read an excerpt as we're waiting for people to ask some questions. And so this is about the immune system. I just ended about speaking about vaccines, and this is from page 142. What I think that is so exciting is that this may drive next generation treatments. And so the four of us write about this, and we call this empowering the immune system to fight Parkinson's disease. So let me just read just a bit for you. Immunization, the same tool that we use to fight infectious diseases, is now being tested as a possible treatment for Parkinson's. When we are infected with a virus, such as chickenpox or the flu, our immune systems produce antibodies to kill it off. Our immune system also reacts to foreign proteins. This is convenient in the case of Parkinson's because misfolded forms of the alpha-synuclein protein, that major factor in the disease, could be targeted. Unfortunately, the human immune system, for uncertain reasons, does not clear these bad misfolded proteins. Scientists are now experimenting with ways to immunize people with the disease so that the misfolded protein is removed, or at least does not spread. Now, before polio vaccines were developed to prompt people's immune systems to make their own antibodies, a guy named William Hammond of the University of Pittsburgh had a different idea. He wanted to give individuals existing antibodies against the polio virus to see if they would protect against future infection. Hammond first obtained antibodies from people who had been infected with polio and recovered on their own. He then injected these personalized fighters into children who had become infected. The children received modest benefits. Fewer of them got polio than those who were not immunized. However, Hammond knew the approach had its limitations. The antibodies in short supply and insufficient to immunize an entire community. The other issue was the effects tended to wane, so repeated injections were required. So when Jonas Salk and Albert Sabin developed the effective vaccine, Hammond's treatment actually fell out of favor. In October 2018, researchers tried Hammond's approach against Parkinson's. they gave 80 people 3 monthly infusions of an antibody. This antibody was directed against these toxic misfolded forms of the alpha-synuclein protein. The antibodies did lower the level of these proteins in the blood and in the fluid surrounding the brain where it matters the most. Many companies are now pursuing this approach for Parkinson's. While promising, the approach requires much more investigation. As with polio, vaccines against these malformed alpha-synuclein proteins is an exciting prospect. Vaccine studies have recently begun. In 2017, an Austrian biotechnology company called Afris announced that in an early study of 36 people with Parkinson's, its vaccine did not result in serious safety concerns and was, quote, tolerated well. The vaccine also resulted in the production of antibodies against alpha-synuclein, the bad protein, in about half of the participants. However, the antibody levels did decline over time and required later booster injections. Sound familiar? Sounds like the polio story. These results are encouraging, but much more work remains, including a demonstration that the vaccine reduces symptoms in those with the disease. So that's just a short excerpt about some of the cool things that we talk about, all of these therapies that are coming. And so I'm going to move on here to the live questions. And first, I have a question from Sheila. Sheila is coming to us from Arizona, from Tucson, Arizona. Welcome, Sheila. Sheila wants to know, can you address Duopa and the newest trials on the subcutaneous administration of levodopa? Okay, thank you so much for the question, Sheila. So when we talk about Duopa and subcutaneous dopamine, what we're talking about are ways that we can give that same pill. Okay, so we were giving a pill. Remember, I showed you a picture of what was the yellow pill that is the most effective therapy for Parkinson. That is a dopaminergic therapy, okay? So when we talk about Duopa, Duopa is a gel formulation that we give into the stomach that is basically dopamine, okay? And so this is a promising way to reduce things like turning on and off, so these so-called on-off fluctuations that people have. It's very promising for mild and sometimes moderate dyskinesia and can treat many of the symptoms of Parkinson, but it doesn't stop disease progression. And so the newest trials that have been done show that there are several hours of improved off time with Duopa, and that means that when you have Parkinson disease, it's just absolutely interesting and intriguing that when you take the dopamine, you actually turn on, and things like tremor, stiffness, and slowness get better. And so when you're off, you have these bad symptoms, and tremor, stiffness, slowness, and then sometimes you can be wearing off and coming on, and we call that motor fluctuations. And so Duopa addresses that and improves that by several hours a day, similar to deep brain stimulation, maybe not as powerful on some of the dyskinesias. And then it also requires a tube that goes into the stomach, but a very good therapy for patients. The subcutaneous administration of levodopa, now that is very interesting because for many years, because levodopa is an ethyl ester, okay? And we think about the makeup of that, it's very hard to get it to cross the skin. So we saw dopamine agonists, right? The rotigotine patch and other approaches that go across the skin. But getting something subcutaneously to get across the skin, in terms of levodopa is much more difficult. But the early studies, the early sort of safety studies, we're waiting to see more data on this, look promising. Could be very cool because remember, people with Parkinson, if you ask them to hold out their hands and put all their medications into their hands, they can't fit them into both hands. I mean, that's a lot of medicine. So when we think about, these new technological ways to have patches and to have ways that we can deliver the medicine by Duopa so you don't have to be taking medicines all the time, this is very powerful. And that's my last point. When you use Duopa, it does require a tube. And when you are going to use the subcutaneous version of the dopamine, if it makes it through its trials, which is early days now, you may need less medications. And for the people on Duopa in general, they don't take Parkinson medications during the day, only during the night. Thank you. Fantastic question from Tucson. So let me move down the line here and first say Laura Crawford, just a shout out to you in Florida. Says, great book, consider me committed to the pact. So thank you, Laura. That's an easy question, and we just need as many people as possible to get on Amazon and get the word out and share about take the pact. We got to take the pact. Okay. Gerardo Torres, thank you, Gerardo, for your question. And I don't see where Gerardo is from, but he asks, how can we get Parkinson's disease to get more funding? Seems like HIV already has a robust treatment available, yet the condition remains more well-funded than others. Wow, great question, Gerardo, and you are spot on. I mean, I just couldn't have said it any better. And it turns out that as a disease, we showed you it's the most rapidly expanding neurological disease. It's growing more rapidly in Alzheimer's. There's more cases of Alzheimer's, but this seems to be growing more rapidly. And we got to get on top of this thing, or it's going to bankrupt us and cause all sorts of morbidity and mortality for people, meaning that people are going to get falls fractures, nursing home placement. They're not getting adequate treatments, and we're not moving the therapies along down the road. And so what do we need to do? Well, if you read in the book, we talk a lot about the stories of polio and HIV, and they got out there. And polio, March of Dimes, right? They mailed dimes to the White House, right? They had more dimes than they knew what to do with. And the White House actually drove a fundraiser, and that's where that foundation came from. It was Franklin Delano Roosevelt, and there are lots of great pictures in the book, and that story is just a great story. We haven't done that in Parkinson's disease. In HIV, they occupied the FDA building. They agitated. They were charismatic. They advocated. I mean, just were amazing. And I'm not saying that a lot of you aren't amazing, because there are a lot of great, amazing advocates. That there are so many people. And at the beginning of the book, Ray Dorsey tells this story about how he's giving a talk to the Parkinson's men's group in Rochester about health. It doesn't have to do with Parkinson. And he asks, how many of you know somebody with Parkinson or have somebody with Parkinson in your family? And the entire group raises their hands. So we got to get loud. We got to agitate. We got to be more charismatic. And so, great question. We're not doing it. We're not getting it done. And we got to take a lesson from polio and from HIV. So thank you, Gerardo. That's a fantastic question. I have a question from Chaim Kramer, and it's another, just, you guys are asking really, and ladies are asking really just amazing questions here. Should all patients get genetically tested with results stored in a national or worldwide database? Privacy concerns aside, is this a good idea for helping facilitate precision medicine? So, it turns out that that people are beginning to make these investments, and the Parkinson Foundation has a gene project investment where they're actually paying for people to get tested at centers of excellence and trying to get as many people genotyped as possible. The Michael J. Fox Foundation has been getting into this area as well and trying to help to get people genotyped, and there's lots of now funding for this. Now here's the two sides of this. One, we don't have the therapy yet for people, although we do have clinical trials that are going. Remember, I showed you the two most common genes are GBA and LRRK2. So if you knew you had those, you could enroll in some of these exciting trials, and we need people to enroll in trials. And I should have mentioned that in the presentation, that there is a lack of clinical trials participation in our Parkinson community. And I'm not saying that a lot of you don't participate, because a lot of you are awesome, but we need more if we're going to move these therapies faster. Now, in terms of having national databases, Matt Farrer is a famous geneticist who just joined us here at the FixCell Institute. He loves this. And, if you ask him, he would say, yes, everybody get your genes in here as much as you can. But there are privacy concerns. We need to be sensitive to those. And the more people that we have genotyped, the more we're going to be ready to administer therapies. But also, the more blood and the more DNA we have, we can drive therapies, because we need access to that to develop the new therapies. And so it is a good idea. It is complicated, and we don't want to, to get in the way of people's privacy. So it's just a fantastic question. So I have another comment here from Laura Crawford, who says, regarding the polio story, today we celebrate Dr. Jean McNamara's work on polio. So thank you for that comment. And we are moving here down along. I'm going to check into our QA section. These are comments from our chat. I gave you a bunch of comments from there. I'm really just absolutely stunned with how much you guys are giving us, fantastic feedback here. Love the Parkinson Foundation pin. Thank you. I do support all the Parkinson foundations. I'm the medical director for Parkinson Foundation. So thank you to an anonymous attendee. They're a great organization, as is the Michael J. Fox Foundation, Davis Finney, APDA. These are all just great people. Parkinson UK. If I had time, I would name them all, and they're all in the book. I mean, fantastic. Here's a question from Shabir Latif. Shabir wants to know, what about physical activity and using neuroplasticity? Wow, great question. If we put animals on the treadmill, Shabir, we actually get these neuroprotective disease-modifying changes from exercise. So we are super interested in this in the humans. And what we lack is that biomarker to be able to measure whether or not we're actually having a meaningful effect, okay, on the disease itself. And it can be tricky when an intervention like exercise also has a symptomatic benefit. So if you have a symptomatic benefit, how do you know you're getting neuroprotective benefit? So it takes large numbers of people in studies like that. And so the development of biomarkers, I think, is something that could be to think about. But there's a lot of people, Shabir, who think that exercise is neuroplastic. An anonymous attendee asks, what new therapy are you most excited about? As you can tell, I'm excited about everything. I'm a glass full, half full kind of guy. But I really think that the therapies, the immunotherapies are really cool, and inflammation and immunity are going to be a very new and interesting area. There's a great article in JAMA Neurology about TNF alpha drugs. These are drugs for things like arthritis and autoimmune diseases, and these sort of anti-inflammatory approaches may reduce risk of Parkinson. And so these as therapeutics are going to get looked at. I think all of these new, my lab does all these cool, I think it's cool, but it does all these cool, techno innovations. So I think a lot of the neuromodulation stuff is really cool, and next generation neuromodulation therapies. And I think optogenetics and temporal interference and using DREADS and optogenetically inspired therapies are super exciting. And then all these drug targets, that are unexplored. There's so much unexplored area. So if we can increase our funding by 10 times, think how much more we can explore, and what a great job we'll be able to do. So thank you for that question. I'm super excited. Of course, my lab does the neuromodulation, so I guess I have to. If anybody from my lab is watching, I have to say the neuromodulation and the DBS, because that's what my lab does, but it may not be true. I'm excited about the others too. Yuma Bev is asking us about Parkinson's disease. Welcome, Yuma. Parkinson's disease for over 25 years. Congratulations, that's fantastic. And had bilateral STN DBS about 8 years ago, and is doing fantastic. Asks, is it possible we don't need to keep taking Sinemet? The answer to that is, we really don't know, but we do know now from a few decades of deep brain stimulation that almost everyone, 99.9% of patients, will benefit from being on both the Sinemet and the dopamine along with the DBS. And one of the things that I've seen that's absolutely tragic, Yuma, is a lot of patients in my practice come and they try to avoid dopamine, because early on in the DBS story, people were trying to get patients off of all of their dopamine. And this turned out to cause apathy, depression, walking problems. And so it's kind of like if you were climbing a mountain like Mount Everest. would you keep climbing that mountain and do it without the oxygen tank? Like, or do you want to get to the next level, to the next base camp on Mount Everest without oxygen? The idea of putting STN-DBS in, or GPI-DBS, or whatever brand you use, whatever company, is great. But don't forget, long term, this disease goes for a lifetime and many decades in a lot of patients that we've been studying. So don't forget about adding back in dopaminergics. And so I recommend to all patients that in a lot of cases, people have forgotten about that, and you could be doing even better. And it's not going to increase your disease progression by taking that. So the combination could get you a little bit higher on that mountain if you were climbing Everest. And so great question. I really appreciate you asking that. Amanda Ribello, welcome, Amanda. Amanda wants to know what I think about using levodopa as the first drug for treatment of Parkinson's disease. Thank you, Amanda. Amanda, we just wrote about this. Melissa Armstrong was the lead author. She and I wrote a review for the Journal of the American Medical Association, also called JAMA. And there's a podcast, and we talk about this in the podcast. It's free. So if you want to go online and check that out on JAMA, the answer is we've looked at this, and it turns out that many years ago, there was a movement to not use levodopa and Sinemet early on in Parkinson's disease. And we now know, and we learn, right? We're humbled. I tell my students in fellows training, I know less about Parkinson's disease today than I knew when this whole thing started, for me a couple of decades ago. And so we all learn, and what we've learned, and there are many studies, the most prominent is a British study that appeared in one of the Lancet journals looking at this question. And early levodopa is okay, and it's okay to start with levodopa as the first treatment. So thank you so much, Amanda. That's a fantastic question. Here's a question that we have. I want to participate in research, but I don't know where to look. What are your suggestions? And can I participate from home with COVID? Okay. Well, first of all, if you have COVID, my heart goes out to you. And stay safe. Be phoning your doctors and your healthcare team with regular updates, so they know what to do and they can tell you what to do in case you need to come in. They can be ready for you before you leave your house. They need to know if you have a COVID infection and Parkinson, so they can be advising you regularly. And so I do have patients in my practice with COVID, and I'm checking on them every day if I have them in their house. And it's more complicated once you get the Parkinson plus the COVID. So thank you for that part of the question. And I hope that the people who are listening to that keep your healthcare team informed that you have COVID, and keep asking them what to do and what's the best approach. If you don't have COVID, lock yourself in your house, take your deliveries from outside, have them leave them on the doorstep. You can throw away the bags, wipe things down, although there's still not a case reported by the CDC from food delivery. But this is respiratory. It goes from person to person. So the best way to avoid it is really isolate yourself for the next 4 to 6 weeks. It's doubly important whether you have young onset or later onset Parkinson's disease. Where can you look as the second part of your question? Where can I look to get involved with research? Well, clinicaltrials.gov is a publicly available website that you can look and search for terms for different studies you might be interested in. If you type in Parkinson, you're going to find a slew of studies, okay? And you can do all sorts of search terms. And so that's a really important site. You can look at the foundation websites, Parkinson Foundation, APDA, Michael J. Fox Foundation. And then Michael J. Fox has this really cool Fox trial finder that Todd Scherer, who's one of the co-authors, and his colleagues developed, which is fabulous. And so thank you for that question. I think that was terrific. Let me address another question that just came in, and it builds on the last one. What can a Parkinson patient do to strengthen their lung function to help guard against a bad COVID-19 outcome? Whew, just great question. And we actually don't know the answer to that. We do know that that Parkinson patients do have some problem with their lung function, and we know this from recently, and we have seen from a study that was done, the CVT study on the inhalable form of dopamine, that's sold under the name Ambrosia. And so we know this from that the lung function is abnormal. But actually, if you look back, one of my old colleagues, who's now the dean at Jacksonville University in Florida, Chris Sapienza, showed this in a series of studies many years ago. But we have a lot of data now that shows the lung function is affected. Some people use incentive spirometers to try to get their lungs and their cough to be stronger, and it may be your cough that's more important. And Karen Hegland has a National Institutes of Health study where she looks at cough, and so we don't know if any of these things are good for preparing you to get your systems in order. Although there's one therapy that was shown to reduce aspiration pneumonia. It was published in the journal Neurology with Michelle Trichet, who is at Columbia University. And that study looked at a device called EMST, or expiratory muscle strength training. It was originally invented by Chris Sapienza and Danny Martin and Paul Davenport. And that helps to reduce aspiration pneumonia. But whether it'll strengthen your lungs up for COVID, we just don't know. So just try to stay isolated and be preventive, the big P on that one. So here's another question about Parkinson research funding being so underfunded, and based on what criteria foundations choose which disease to sponsor. Great question. We just believe, and we studied this in the book, and we totally believe this idea that the underfunding of Parkinson comes because we haven't been strong enough in our advocacy. We're not agitating enough. We're not being charismatic enough. We need to push, push, push. That's why we wrote this book. That's why we need you to share and talk about taking the pact. We're only going to do this if everybody on this call, on the grassroots, we got to get down to grassroots and bottom up the pact. I mean, if we don't bottom up this pact, get on Amazon, talk about the book, talk about it on our social media channels, talk about the pact. We got to be as aggressive as putting a condom, like they did in HIV on one of the Congress members' houses. We got to be aggressive. We got to occupy the FDA building if we need to. I'm not saying run in and invade the FDA. Okay, that's probably a little bit much, but they did that in HIV. We're not doing that. So if we're going to go by a factor of 10 times or more, then I think it's super, super important. So we have a question about many of the therapies being super expensive, especially with all the medications or high-tech delivery systems that are used. How should our healthcare system adjust to reduce the cost on all involved? Fantastic question. We actually address this in Parkinson's disease book. It's really tricky because there's a lot of advocacy going on, behind the scenes here. This is really where the Parkinson Action Network, which is a network that works together through the Michael J. Fox Foundation, but all the major foundations participate together and try to come with a singular voice to try to push the legislation forward. Now, we've heard that there's bipartisan support. Okay, so in this time where we're so divided, okay, I'm happy to say when it comes to research and research for Parkinson, and when it comes to thinking about these questions, we're not divided. We are not bipartisan. Everybody knows what we should be doing. And so we have supported the National Institutes of Health as a bipartisan effort. And every time they've tried to chop it the past few years, the senators and representatives have come together. So we can be proud about that. We just got to increase the amount of funding by a factor of 10 or more. And we've got to get the bills passed that are out there that are going to reduce the big pharma costs. Now, we do need to acknowledge we can't do this without a partnership with big pharma to get these drugs out there, and with device companies to get devices out there. But we've got to make it more affordable for patients, and we've got to create a system where everybody is gonna win at this. And so thank you so much for that question. That's a great question. I want to move on to
Michael.
Yes.
We are at the 7 o'clock mark already. I can't believe how quickly that flew by. We have questions, which we can address over email, and make sure to post those responses on our website as well.
Fantastic. I can't believe the hour flew by, Abby. It went by very fast. It was a lot of fun.
Thank you so much, Michael. I'd also like to thank the Parkinson and Movement Disorder Alliance for helping to promote this talk and the book in general. I also want to note that the University of Rochester has a neurology study interest registry, and many of our studies are virtual, so it's possible to learn about research, potentially participate from the comfort of your own home. So that's something we'll make sure to put on the website as well. But feel free to reach out in the meantime to info@endingparkinsondisease.org if you have any questions.
Thank you very much for having me, and I'm going to answer all your questions on the Q&A as well. So have a great night. Take the pact, everyone. We appreciate you.